So the chemical structure of DNA is, first, it contains a phosphate group connected to a sugar, which is connected to a nitrogenous base. These bases can either be adenine, guanine, thymine, or cytosine. There are two different ends to the nucleotide. The first end is the 5' end, and the second is the 3'. The difference is the 5' end has a phosphate group attached to it, and the 3' end is just a hydroxyl group, and that's where new DNA will be attached to the 3' end because it doesn't have another phosphate group. So we can say that DNA travels from 5' to 3'. Watson and Crick discovered that DNA was a semi-conservative model, meaning that the duplications of the DNA would contain 1 part original DNA and 1 part copy, shown by the purple being the original and the red being the copy. So DNA is split by the enzyme helicase, which causes the 2 separate strands, and one being the leading strand and one being the lagging strand. After the helicase splits, the strands, binding proteins are added to or attached to the strands to keep them in place so they don't bind together again. On the leading strand, it's called the leading strand because everything works smoothly because the DNA is naturally traveling from the 5' end to the 3' end. So the DNA polymerase just travels down, adding on new strands, and you can see the red is the new and the blue is the old, and that's just the 1 copy of the DNA. The leading strand doesn't have any problems. It just goes because it's from 5' to 3'. It doesn't need anything changed to it. And the DNA polymerase is the actual part that's creating the new strands of DNA. Now, the lagging strand has some problems because it's actually moving in the opposite direction. So it's going from 3' to 5' when it needs to go from 5' to 3'. First, we have our DNA primase, which synthesizes RNA primase, which we will need because RNA is actually produced. And we still have DNA polymerase, which will still create the new copies of the DNA. So what happens first is the DNA primase joins the RNA nucleotides into a primer, which is the red part shown. Then the DNA polymerase adds new DNA nucleotides to the primer, and this part right here is called the Okazaki fragment because that's the name of the scientist who discovered it. Then after the DNA polymerase reaches the next RNA primer, this part, it will detach. Then the DNA polymerase reattaches again, creating new nucleotides until it reaches the following primer. And this process continues until the strand is completed. If a DNA strand ever unwinds too much, there's topoisomerase, which just rewinds the strands that don't need to be unwinded. And lastly, in the lagging strand, the Okazaki fragments are all connected through or by DNA ligase. And because since there's 2 different strands, one lagging and the leading strand, we say that they run anti-parallel, even though they are lined up straight like this. One runs 5' to 3', while one runs 3' to 5', so they're opposite or anti-parallel.